Now here you see we will be doing, here it is written pistil, megasporangium, ovule and the embryo sac, and this is what we will be taking up today. Now you see we will be dealing with gynoecium, that represents the female reproductive part of a flower, and that is a single pistil, that means it is monocarpellary pistil. They have got one single carpel. Then we have other fused pistil, that is known as multicarpellary, and when there are more than one pistil that may be fused together, we call it as syncarpus, as in case of hibiscus, and if they are free they are known as apocarpus. Okay. Now every single pistil has got stigma, style and the ovary, and this ovary presents the ovular cavity, that is known as locula. And what is placenta? Placenta is a lining into which the ovules are attached. Okay. And here you see the diagram in this figure. You see stigma, style and the ovary. Okay, thalamus from the base in which they are attached. Now here you see, here it is syncarpus, it is exactly of that of a hibiscus, they are fused together. Now here you see carpels, they are fused, they are not fused, they are free. Okay, so that is known as apocarpus, if they are free. Apocarpus or multicarpellary, you find it in custard apple. Now this diagram, you see, this is a very important diagram, and that will be here. Here you see we have, here it is written the embryo sac, that means this is exactly the structure which ultimately, and this is the embryo sac, this embryo sac that has the ovules in it. Okay, that later develops into a seed and is capable of giving rise to a new individual. Now if we see this diagram very carefully, you see we have got an outer integument. Okay, outer integument is there, then after that you have the inner integument, right? Then here you have a nucellus. This part is nucellus, and here this is the embryo sac. Now nucellus, if we consider, now this is having the reserve food material. Okay, now if we take a seed, if you see the seed, you see here you get a point of attachment or a scar like structure. This is actually the hilum, and this is the point which shows the point of attachment. Okay, and then here you have a small hole through which, if you just soak a bean seed or a gram seed, you see air bubbles at that. So this is the micropilar end. Okay, this is a very important, and this forms the area through which air aperture enters, point of attachment and air aperture. Now this structure we will be dealing when we will be dealing the fertilization section. So here this entire structure is known as megasporangium, and we will be dealing with the process of megasporogenesis. Now what is megasporogenesis? Megasporogenesis is actually the process, okay, it is actually the process that leads to the production of ovule, that is known as megaspore mother cell. Okay, now megaspore mother cells. Now you see, here the gynoecium is generally in plants, it is known as monosporic. Why monosporic? Because it is only one spore that is capable of fertilization. Okay, now what is the division that undergoes here? It is the meiotic division. Okay, it is a meiotic division undergone by the nucellus or the reserve cells like that. Now this nucellus, you see, first the entire thing has got single one cell. Okay, now it first divides. Now again it divides into two. It is double. Now each double divides into 4, and that is known as a tetrad condition. Okay, after that tetrad, each divides into 2, so you get an 8-celled stage. Okay, now this, when you get, I'll show you the diagram. Now see, 8-cell stage, in that 8-cell stage you should remember one thing, that 6 are enclosed by the cell wall. 6 cells are enclosed by the cell wall, and 2 cells occupy the central position, and that is known as the polar cells, okay, or the egg cells. Now you see here, You see, you have a single cell that is known as the, see, nucleus cell divides into megaspore, mother cell, single cell. Then from the single cell, you have dyads. Dyads means two cell stage. You see here, the upper part represents the micropylar end. Clear? And then here, it is again the tetrad, 4 cell stage. Now from there, 2 cell stage, 2:4, 4:8. So this is how the meiotic cell divides. And you see, I said that out of that, 6 are enclosed within the cell wall. You see the upper part, the Cinegrid part. Okay, Cinegrid and egg cells, the central 2 cell stage, and 3 antipodal cells. Okay. Now you know this, the chalazial end, when we were dealing with the earlier ovule, it was right at the bottom. But you see, somehow it gets upside down. See, base here, these cells, you know, divides, and that suspension cell gives it a twisted structure, so that it gives a hanging thing. You know, this goes up and these goes down. So we have the Cinegrid cells at the top. You see here the antipodal cells, they are attached. This is a chalazial end. Then in the center, you have the polar nuclei and the central cell. Okay, you see, polar egg cell, central cell, and here, you know, this again, that Cinegrids. Here, see, Cinegrid was here, again Cinegrid has come down. So here Cinegrid, you have filiform apparatus. What is filiform apparatus? Filiform apparatus is actually, it guides the pollen. Guides the pollen grain, the nucleus of the pollen grain, that is the generative nucleus, to the egg. So this is why filiform is important. Okay, and here these are stored food. So these part we will be dealing when we will be dealing with the fertilization part. Here you see the 4 nucleus, then we have got 8 nucleus state. Then this is the female gametophyte, the embryo sac is there, central cells, egg apparatus is there. Okay, 2 Cinegrid cells are there, egg cells, antipodal, like this. Okay, now next topic, what is this? Is pollination. What is pollination? Now pollination, it is a mechanical process of transferring of the male gametes to the female gamete. So this particular and which is again, you know, pollination is a pre-fertilization event also. After pollination, fertilization events occurs. Okay, so there are very different kind of pollinations. There are various kinds of pollination that we will be dealing in our next class.